Hello everyone, my name is Meredith Frazier and I'm a second-year postdoc in Robin Stanley's lab at NIEHS. I'm really excited to share my work as part of the NC State Postdoctoral Research Symposium. Today I'm going to tell you about a COVID-19 viral protein and its search for U. Not U, but the letter U, and the RNA code that makes up the virus. Viruses in the immune system are in an evolutionary arms race, each trying to gain an advantage over the other one. I labeled the protein I'm studying, Nsp15, as the sword in this central image, but it might be better to think of it as an invisibility cloak because it helps hide the virus from immune system detection by cutting up excess viral RNA. Because of this function and the fact that it's found in all coronaviruses, Nsp15 could be a good therapeutic target, not just for COVID-19, but for other coronaviruses as well, including ones that cause the common cold and ones that have caused epidemics in the past. While the new vaccines are very promising, with hundreds of millions of cases and millions of deaths globally, this pandemic highlights the need for effective treatments against coronaviruses as well. However, we need a better understanding of how Nsp15 specifically finds and cuts the RNA code to be able to design a drug for it. To tackle this objective, we took a structure-function approach, asking, what does Nsp15 look like at atomic detail, and how does it work on a molecular level? We took pictures of Nsp15 using cryo-electron microscopy to build models of Nsp15 in different reaction states. These structures helped us answer questions about how it specifically recognizes U's in RNA, how it cuts the RNA, and why it does so not just with one copy of the protein, but with 6 copies working together. Looking at our structures of the protein with and without RNA bound, and running computer simulations on it, so far suggest a very flexible Nsp15 copy is stabilized by forming a complex with other copies of itself, but it still wobbles about until it finds a U in RNA, which binds and fixes the protein into place so it can cut the excess viral RNA, ultimately helping the virus stay hidden from our immune system. We turned to biochemical assays to study how Nsp15 cuts RNA. Our structure showed us parts of the protein, specific amino acid residues, that may be important for cutting RNA. One question we asked was if we change those parts, does it affect how Nsp15 cuts RNA? Our functional activity tests on these versions of Nsp15 allowed us to confirm their importance that we had hypothesized from our structures. Another question we asked biochemically was, does it have any other preference for cutting RNA other than seeing the letter U? Our data shows us that Nsp15 is mainly guided by the presence of a U in the RNA, although the letter after the U does matter somewhat, with it preferring an A or a G. Our structure-function approach helped us understand how the SARS-CoV-2 viral protein Nsp15 specifically recognizes U's and how it cuts the RNA. Now we are interested in what other factors may affect how Nsp15 carries out its immune-evading RNA-cutting action. Are other viral proteins involved? Does the structure of the RNA matter? This work will answer basic biological questions about Nsp15, which we hope will ultimately lead to new therapeutics. Thanks for listening.